Aquaria is a new web resource designed to help biologists use 3D structures to gain insight into protein function. To use Aquaria, we first specify a protein. In this case, let's choose P53, a protein implicated in many human cancers. After we specify a protein name and organism, Aquaria displays basic information about the protein, as well as a concise visual summary of all matching structures in the PDB with significant sequence similarity to the specified protein. For P53, we see that Aquaria finds 120 matching structures. From these, Aquaria automatically determines which structure has the largest number of identical residues when aligned onto the full-length protein sequence. This structure is then displayed at the top of the screen, and details about the structure are shown in the right-hand panel. The molecule is initially colored to indicate secondary structure elements, but this is augmented by dark coloring for residues in the structure that differ from the specified protein sequence. Clicking on one of these darkened residues lets us compare the amino acid occurring in the structure with that in the wild-type P53 sequence. We see that, in this structure, many residues differ from the wild-type P53 sequence that we initially specified. Furthermore, when we look at the overall alignment of the structure onto the full-length P53 sequence, we see that a 30 residue region has been deleted, fusing together two domains by removing a linker region. Because we've seen that the structure has so many changes from wild-type P53, we may be more interested in other structures that have fewer changes. The matching structures section helps us here, since it ranks structures by sequence identity to P53. Let's look at this group of 87 structures. Clicking on the number opens a tree view where the structures are grouped by molecular configuration. We see here, for example, that 45 of these structures consist of a single molecule of P53 by itself, while other structures have P53 in complex with a range of different molecules. Let's choose a structure containing a tetramer of P53 bound to a dimer of DNA, which is believed to be one of the most important physiological states of P53. When we look at a complex structure such as this one, which contains many different molecules, Aquaria simplifies the view by automatically focusing on one molecule at a time. We can escape this mode by double-clicking on the background, thus showing all molecules in the structure, or switch the focus to any other single molecule just by clicking on any part of it. In addition to displaying 3D structures, Aquaria also allows mapping of sequence feature information. Clicking on the Features tab accesses a range of interpro and UniProt features, and clicking on a feature lane changes the molecular coloring, in this case highlighting the residues of P53 that are known to undergo a variety of specific post-translational modifications. In cases such as this, where we may need more screen space to examine details of a 3D structure, it can be useful to activate full window mode available from the title bar. The title bar also gives a summary of sequence to structure information currently being displayed. In this case, it reminds us that we're looking at the P53 sequence, which we first specified, aligned onto a particular PDB structure that matches with 100% sequence identity. For most proteins, however, the sequence to structure alignments will usually have a much lower identity. To see how Aquaria deals with such cases, let's switch to another protein. Let's choose GLUT4, a key player in diabetes for which structural information is currently only available via homology. Notice that now many residues have darkened color, since this structure is derived from a different but related protein. In general, as the relationship between sequence and structure becomes more distant, the number of dark residues increases. This provides an immediate visual indication of the quality of the sequence to structure alignment. Further details in the alignment quality are also available from the title bar. A key advantage of Aquaria compared with similar systems is that for each sequence, it allows us to easily explore all related 3D structures, often including many that are only very distantly related to the protein we initially specified. For example, of the 6 structures represented here, the top-ranked structure is only 16% identical to GLUT4. Clicking on the group loads the top-ranked structure, which we can then map with features from GLUT4. For example, let's highlight the extracellular and cytoplasmic parts of this transmembrane structure to provide an overall orientation. We can now see that this structure is in a state known as outside open, during which sugar molecules outside the cell are believed to bind. In contrast, some of the other matching structures show the inside open state, during which sugar is then released into the cell. Thus, by exploring the range of states and molecular interactions available in the matching structures section, we can gain detailed insight into the molecular processes underlying protein function. Aquaria also makes it easy to select regions of interest just by clicking while holding down the Shift or Control keys. At any time, we can then zoom to the current selection by pressing the Enter key. We can also do standard operations on the current selection, such as changing color, or by using the Copy command, we can then paste information about the selected region into a text document. The Aquaria Help Facility provides basic pointers for getting started, as well as detailed help with installing Aquaria. You can also access a summary of all mouse and keyboard commands, and you can also access information about Aquaria's API, which, for example, allows mapping of user-defined feature sets. Aquaria has been designed to make structured information easier to access and use, freeing researchers to focus on understanding and interpretation, and thus helping accelerate discovery in the life sciences.